we chose was equine mesenchymal stem cells from bone marrow, adipose tissue, and umbilical cord. Immunophenotypic characterization and differentiation potential. This is presented by Melissa Nowick, Kimberly Weller, Norman DeWitt, Bradley Metz, and Cecilia Gamian. Mesenchymal stem cells, commonly referred to as MSCs, are progenitor cells that can be easily isolated from various adult tissues. MSCs are non-hematopoietic. This means that they make up a small proportion of the stromal cell population in the bone marrow and can generate bone, cartilage, fat, cells that support the formation of blood, and fibrous connective tissue. They don't function primarily in forming blood or blood cells. They also are multipotent. This means that they have the ability to develop specific types of cells that are terminally differentiated. They also have extensive proliferative ability. This means that they can grow or multiply rapidly. And finally, they have the ability to differentiate in vitro into different lineages, such as osteoblasts, adipocytes, chondrocytes, tenocytes, which produce collagen molecules and tendons, myocytes, neurons, glial cells, and hepatocytes. Mesenchymal stem cells may be isolated from different locations in the body. These include bone marrow, which is the most studied and used source in equines. Also, adipose tissue. It is the most abundant and accessible source for mesenchymal stem cells. Also, the amniotic membrane and umbilical cord. It is less immunogenic, meaning that it's less likely to produce an immune response. Non-invasive, meaning that when you extract the cells, you're not entering or penetrating the body to disturb body tissue, and they can differentiate into nerve and endothelial cells. The use of mesenchymal stem cells has been demonstrated into the cartilage, bone, and tendon of horses. Equine mesenchymal stem cells are identified by their adherence to tissue culture plastic in the lab and their ability to differentiate into multiple lineages. Immunophenotyping, the analysis of heterogeneous populations of cells in horses, is prevented by three things. First, a lack of specific cell markers. Next, a limited number of monoclonal anti-horse bodies prevents immunophenotyping. Monoclonal anti-horse antibodies evaluate the immune responses in horses and target important antigens of equine infectious agents to which protective immune responses are directed. And finally, evidence that certain markers of other species do not cross-react with equine species prevents immunophenotyping. Several cell markers, positive and negative, have been tested and used to identify other species' mesenchymal stem cells and adipose-derived stem cells. These include the CD44, CD90, CD105, CD34, and MHC2 markers. Mesenchymal stem cells have been proven to improve regeneration and tissue function. Autologous therapy, when MSCs are extracted from the individual, are used in therapy for the individual themselves, is widely used. Its benefits are that it doesn't result in any harmful effects or inflammatory responses in the individual. It's limited by two things. The expansion of MSCs by culturing takes 10 to 21 days in the lab. Also, in elderly people, there's a decrease in the quantity, proliferation, and differentiation potential in MSCs. Allogenic treatment in horses offers advantages in acute injuries. Allogenic treatment is when the stem cells come from another organism of the same species, so another horse. MSCs can be injected quickly into the horse, which eliminates the time needed for culturing of autologous MSCs. The lack of MHC2 on the cell surface is an important factor in determining whether MSCs can be used for allogeneic treatment. The lack of MHC2 gives the MSCs the ability to escape T cells and prevent an immune response. The study assesses three different aspects of mesenchymal stem cells. It assesses the differences in cell culture of bone marrow, adipose tissue, and umbilical cord tissue stem cells in equines. It also determines the immunophenotypic characteristics three aspects are compared for research or for an allogeneic therapy cell bank in this study.
Method section. This just provides information by which a study's validity is judged. It offers clear and precise description of how an experiment was done. For bone marrow, it was collected. Bone marrow was collected. MCS was performed by aspiration. 5 animals aged 7 to 12 years old were sedated. Local anesthetic blocks performed in the fifth vertebrae where bone marrow cells were collected. 2 syringes were used for the bone marrow collection. Samples of each animal were centrifuged for 10 minutes and the supernatant was discarded. DM-EM low glucose and histopac were added at a ratio of 1 to 1 and centrifuged for 40 minutes. DM-EM low glucose was then added again, but it was only centrifuged for 10 minutes, twice. Cell number was determined using the Triton blue exclusion method. This method just colors the dead tissues and cells blue, so you can tell which ones are which. Cells were then cultured with specific culture medium, which was changed every 3 days until confluence was greater than 80%. Confluence is just the curvature of the plate. Next, adipose tissue. The collection of adipose tissue is done in the region of the dorsal gluteal muscle, which is just the tailbase. Animals 6 to 13 years old were sedated intravenously, followed by inverted L block of the local anesthetic. Intravenously just means that the drug was administered into the vein. Around 1 gram of adipose tissue was collected and stored for 20 minutes at room temperature in a sterile 50 milliliter tube. A sample of adipose tissue was washed 3 times with HVSS penicillin. It was submitted to mechanical separation using a scalpel and anatomical forceps, which is kind of rounded and curved so it doesn't damage the tissue. Then it was placed in a solution of collagenase at 37 degrees Celsius for a half hour. Collagenase is just an enzyme that breaks peptide bonds in collagen. The solution was then filtered through 70 micrometer filter. DM-EM low glucose was added and centrifuged twice for 15 minutes. The sample of cells was stained with Triton blue once again and counted using a microscope counting chamber. This is just a microscope slide that's specifically designed to be able to count cells. Cells were cultured with specific culture medium, which was changed every 3 days until cell confluence was greater than 80% was reached, like before. Umbilical cord. For samples from births, about 10 centimeters of umbilical cord from the fetal portion was collected immediately after delivery and stored in a sterile 50 milliliter tube. For samples from the slaughterhouse of pregnant uteri were separated and opened, and a sample of 10 centimeters of umbilical cord from the fetal portion was collected. Fetus of 9 to 10 months of age were used in this process. Umbilical cord samples were washed 3 times in the HVSS penicillin. They were submitted to mechanical dissection to separate the veins and arteries, which were then thrown out. The periovascular region of the umbilical cord tissue was fragmented using a scalpel and anatomical forceps. It was placed in a solution of type 1 collagenase at 37 degrees Celsius for 1 hour. The sample was processed like the adipose tissue, except the culture medium had DM-EM high glucose instead of the DM-EM low glucose like we saw before. Trypsination of cells was accomplished when a confluence of greater than 80% was reached. Trypsin had to be added at 37 degrees Celsius for 5 minutes. Cell suspension was centrifuged twice for 10 minutes to remove the trypsin, and then cell pellet was resuspended in a culture medium and divided into 2 different flasks. Cells were cultured under the same conditions as before. All of these kind of followed the same methods. They gave different results, but all in all, they did basically the same thing for every one of them.
Okay, so the second part of our method looked at different types of differentiation between these mesenchymal stem cells. The first procedure that they did was looking at osteogenic and adipogenic differentiation of the mesenchymal stem cells. So, 3 different stem cell samples were placed in 6-well plates. They were incubated at 37 degrees Celsius in a humidified atmosphere, which contained 95% air and 5% CO2. After reaching 80% confluence, which we said already was the coverage of the plate that it's on, the medium was removed and the differentiation media were added. And these differentiation media were changed every 3 days. Adiabagenic differentiation was confirmed by the deposition of lipid droplets in the cytoplasm with a stain called red O. The osteogenic differentiation, on the other hand, was confirmed by the red S staining of the extracellular calcium matrix. The next thing that they looked at was the chondrogenic differentiation of the mesenchymal stem cells. In this part of it, they took a pellet of mesenchymal stem cells, they incubated it in the same conditions, the 37 degrees with 95% air, 5% CO2, and as before, the differentiation media were added when they were removed, and they were changed every 3 days. To confirm that chondrogenic differentiation did occur, the pellets were stained with what they call alkene blue and toulidine blue, and both of these were able to identify the presence of proteoglycans. The third method was called flow cytometry. And in this method, they used a caliber flow cytometer, and they used this to perform an immunophenotypic analysis of 3 different stem cell types. The 5 things listed here, these 5, were the antibodies that were used during this process. And this part of the study was used the protocols that were suggested by the manufacturer, so they followed those instructions very carefully. The 4th method was called immunocytochemistry, and this was done for the MHC2. In this case, samples were plated in 24 well plates, and once again, after 80% confluence was reached, the cells were fixed and permeabilized. Next, an endogenous peroxidase block was conducted, and after the block was conducted, they incubated the samples in a milk powder solution. After 18 hours, the primary antibodies were detected, and the evaluation of the reaction was performed using what they call an inverted light microscope. So as you can see, all these parts were pretty similar, but used different variations to find the different types of differentiation. And one of the most important parts, the statistical analysis, I'll try and put simply. The dependent variables were evaluated with what they call an analysis of variance. In this case, sources of variation in the model included cellular source in animals, which were considered to be fixed in random effects. A transformation was applied to the percentage data, and this was done to improve the normality of the data. And if the analysis of variance was significant, means were separated using the least significance difference. And overall, the data were reported as the least square means.
The mesenchymal stem cells from the bone marrow sample, adipose tissue, and umbilical cord sample were all successful in attaching to the flask in a matter of days. The bone marrow and umbilical cord took 48 hours, while the adipose tissue only took 32. An early connective tissue growth could be seen after an average of 4 days. More specifically, the bone marrow took about 4.5 days, the adipose tissue took around 3.5 days, and the umbilical cord took an average of 4.8 days. For the cells to transition from primary growth to third passage growth, the number of days was higher. Bone marrow took 25 days, adipose tissue took 15.5 days, and the umbilical cord took 26.75 days. The time for the cells to reach 80% cell coverage of the plate for bone marrow was 11 days. For adipose tissue, the average was 7.3 days, and the average for umbilical cord tissue was 15.25 days. After the first passage, the time for the samples to reach an 80% coverage was shortened. The average for bone marrow was 5.2 days, the average for adipose tissue was 3.2 days, and the average for umbilical cord was 6.16 days. The picture to the right shows each of these cells after they reach 80% picture shows the bone marrow, the middle picture shows the adipose tissue, and the bottom picture shows the umbilical cord sample. The osteogenic and adipogenic lineage potential for differentiation was 10 days in bone marrow, 8 days in adipose tissue, and in the umbilical cord, it did not show any differentiation in the given time frame. Because of this not showing, the procedure was repeated. The repeated procedure showed that it took 15 days for the umbilical cord. The osteocytes were confirmed by a positive stain from alizarin red dye. From these samples, most of the cells structure changed from a polygon into a polygon shape. The time for chondrogenic differentiation was 21 days for all samples. In this picture, it shows each of the steps of the procedure and what the cells look like. On the far left, we have the bone marrow cells. In the middle column, we have the adipose tissue cells, and on the far right column, we have the umbilical cord cells. The first row shows the lipid droplets in each sample, and the lipid droplets confer the adipogenic lineage. The second row shows the cells stained with alizarin red, and this stain showed the calcium matrix that it formed. In the third row, after the chondrogenic differentiation, alkane blue stain was used. And then in the last row, it shows the hyaline matrix with toluidine blue stain used. Using immunochemistry techniques, the flow cytometry showed that CD90, CD105, and CD44 markers were all positive, while the CD34 marker gave a negative expression. The MHC2 had a low expression. In the picture to the right, it shows each of these cells after the third passage. The top picture, again, is the bone marrow. The middle picture is the adipose tissue, and the bottom picture is the umbilical cord.
Equine mesenchymal stem cells, or MSC, can be obtained from numerous different sources, but this study chose to focus on bone marrow, adipose tissue, and umbilical cord tissue. When retrieving the MSC from bone marrow, a disadvantage is that it tends to be a more painful process, but an advantage is that it is quick. Adipose tissue is an advantage because of its abundance, but it can be difficult to collect from a horse that is lean. The umbilical cord MSC can be retrieved without causing harm to the foal or the mare. As stated in the results, this study focused on the ability of the MSC to differentiate into osteogenic, adipogenic, and chondrogenic lineages. The bone marrow MSC, along with the adipose tissue MSC, were able to differentiate into osteogenic lineage within 10 days, and it took about 8 days for differentiation into the adipogenic lineage. Umbilical cord differentiation into osteogenic and adipogenic lineage took about 15 days. This results means that although there are differences in the amount of differentiation time, the bone marrow, adipose tissue, and umbilical cord MSC were all able to differentiate. Osteogenic differentiation MSC had the best results for the bone marrow, and chondrogenic MSC had the best results for the umbilical cord and the bone marrow MSC. For this experiment, markers were used. The markers that were used were CD90, CD34, CD44, CD105, and MHC2. The MSC from bone marrow, adipose tissue, and the umbilical cord under culture conditions showed a high positivity for the markers C90, C44, and CD105. CD34 was negative and MHC2 was very low. The MHC2 expression in all samples did not show significant differences, but the adipose tissue MSC had the lowest expression of MHC2, which means that the mare would have a reduced risk of an immune reaction. For this particular study, umbilical cord MSC showed a lower expression for the marker CD90 when compared to bone marrow and adipose MSC. This is thought of because as pregnancy develops, the CD90 marker decreases, and all the samples were collected from mares in their final months of gestation. When obtaining MSCs, bone marrow and adipose tissue proved to be the best when under in vitro conditions. This is
MSC proved to be the best when it came to musculoskeletal regeneration in the horse. This is because of the ability of bone marrow MSC to differentiate. Because of the low expression of MHC2, bone marrow MSC and adipose tissue MSC should be used in clinical trials involving allogenic therapy. Bone marrow and adipose tissue are preferred for cell banking. Overall, bone marrow